The overall goal of this procedure is to collect high-resolution images of photoreceptor proteins using atomic force microscopy. This is accomplished by first obtaining a sample of the photoreceptor and diluting it. The second step is to apply the protein sample to a freshly cleaved and clean mica surface for about 30 seconds. Then, the mica is rinsed with buffer to remove unbound and loosely attached molecules. Next, the sample is attached to a liquid cell that is equipped with an appropriate probe. This assembly is loaded onto the scanner. After filling the liquid cell with buffer solution, engage the tip with the sample and begin scanning. Ultimately, atomic force microscopy incorporating PeakForce quantitative nanomechanical property mapping is used to collect images of single photoreceptor protein on the sample's surface.
Atomic force microscopy that incorporates PF-QNM can answer key questions in the field of molecular biology, such as what are the important structural and chemical characteristics that determine the function of biologically active macromolecules.
The main advantage of this technique over current methods such as NMR and X-ray crystallography is that high-resolution images of intact proteins and other biological macromolecules can be obtained in a biologically relevant environment.
Generally, individuals who are new to this method struggle because the tuning of the probe in solution requires multiple steps, some of which are quite complex. With PeakForce QNM and automated software, several of these steps are made simpler.